Today we start the second chapter, Reproduction in Flowering Plants. All of you know flowers are the modified shoots of the angiospermic plants. A flower has got the main reproductive organs like the endoecium, which represents the male reproductive organ, and the gynoecium, which represents the female reproductive organ. The endoecium consists of a filament and anther. Anther has got, if you see the CS of anther, it has got pollen sac, a line of dehiscence is present, and the filament. The gynoecium or the pistil is made up of stigma, the style, and the ovary. It also consists of the thalamus. The pollen grains, that is the mature pollen grains, they fall on the stigma, and through the style it reaches the ovules. Sexual reproduction in angiosperms consists of three phases: the pre-fertilization phase, the fertilization phase, and the post-fertilization events. Fertilization, all of you know, is of double fertilization. A complete flower has floral leaves arranged in four whorls, that is the calyx, the corolla, endoecium, and the gynoecium. The calyx and the corolla are called accessory whorls as they are not involved in reproduction. The endoecium and the gynoecium are the essential or reproductive whorls as they are directly involved in the process of reproduction. Endoecium is the male reproductive organ which consists of stamens. Gynoecium is the female reproductive organ which consists of the gynoecium is the female reproductive organ which consists of the carpels where female reproductive units are formed. Now students, we come to the stamen or the microsporophyll. The stamens are modified leaves, also called as microsporophylls, and are concerned with the production of microspores which later become the pollen grains. A typical stamen consists of two parts: a long cylinder stalk called the filament and the terminal bilobe structure called the anther. Now, how does our microsporophyll develop? The microsporophyll arises as a small outgrowth called as microsporophyll primordium. On the thalamus, it consists of a mass of meristematic cells. The apical portion of the primordium shows enhanced activities of the meristem and differentiates into anther, while the lower basal part forms the filament. If you see the TS of a young anther, you will find that it consists of a homogeneous mass of meristematic cells bounded by an epidermis. When it grows, it becomes slightly four-lobed. A hypodermal layer becomes differentiated. Its cells are large, radially arranged, and have dense cytoplasm and a prominent nucleus in each. This layer is called the archisporial layer. One cell in the archisporial layer in each of the lobes becomes differentiated as the archisporial cell. It gives rise to the microsporangium of that lobe. This cell divides to form an outer primary cell walls This cell divides to form an outer primary wall cell or primary parietal cell and an inner cell called the primary sporogenous cell. The outer cell undergoes division to form the wall of the microsporangium, while the inner cell, by further division, forms a mass of sporogenous cells which later produce microspores. Now students, we come to the structure of microsporangium. Follow the diagram given. If you see the TS of a microsporangium, it is nearly circular in outline and surrounded by a wall consisting of the following: a single layer of epidermis, a single layer of endothecium where the cells have hygroscopic fibrous thickening. This layer helps in the dehiscence of the anther. 2 to 3 layers of cells forming the middle layer, and the innermost layer is called the tapetum, consisting of large cells with one or more prominent nucleus or nuclei. It is of physiological significance as it nourishes the sporogenous cells and the developing spores. The center of the sporangium is occupied by a mass of sporogenous cells that are characterized by large size, dense cytoplasm, and a prominent diploid nucleus. Later they form haploid microspores by undergoing meiosis. As the anther matures, the two microsporangia of each side fuse to form a pollen sac, and thus a mature anther has two pollen sacs, and hence the anthers are described as diathecious. The two sides of anthers are separated by a longitudinal groove that runs along the length of the anther but are joined at the back by a connective formed by the terminal end or tip of the filament. A singular vascular strand is present in the connective.